Hey everyone, my name is Ian Schrock and this is my Unit 2 Case Connections PowerPoint presentation. So let's just get started. The first case study that I chose was the glucose uptake problem. As you can see here, we have a glioblastoma tumor. We have an MRI and a PET scan of the tumor here. We did learn that there is tissue necrosis within the tumor, so there are some dead cells, dead tissue there, and you can clearly see that on the PET scan. The MRI, we're not really going to care that much about that because we labeled D-glucose with a fluorescent label, and this shows up on the PET scan. Here on the MRI, this is just fluid accumulation and edema around the tumor. But what we clearly see here is this big bright orange blotch, and that is the D-glucose that has been fluorescently labeled. You can clearly see a difference from the surrounding healthy tissue, and we're going to take a look at why that might be. But first, we have to look at the normal glucose uptake in a variety of tissues, and then we'll move into the brain, and that'll come into play with the tumor. We're going to look at skeletal muscle, adipose, liver, and brain tissues. Skeletal muscle and adipose tissue use glucose transporter 4. It is one of the glucose transporter isoforms, but we're only going to talk about the ones relevant to these tissues. Glucose transporter 4, or GLUT4, is an insulin-dependent glucose transporter, and it brings glucose into the cells via facilitated diffusion. GLUT2 is the transporter in the liver, and it is an insulin-independent glucose uptake, and we're going to be talking about why in a minute. This has a high Km and a low affinity for glucose. On the other hand, with glucose transporter 3 in the brain, it has a low Km and a high affinity for glucose. It is also insulin independent, and we're going to talk about the importances of why that is in the brain. The brain also uses glucose transporter 1, but not all cells in the brain use glucose transporter 1. Moving on, in close in-depth look at the insulin-dependent uptake of the skeletal muscle and the adipose tissue. Glucose transporter 4 is coupled with hexokinase 2. Hexokinase 2, if you're not familiar, is responsible for the rate-limiting step of glucose metabolism. It phosphorylates glucose to glucose 6 phosphate. And what's cool about the insulin-dependent uptake, what makes it insulin-dependent, if you can see my cursor here, the insulin binds to a receptor on the plasma membrane of the skeletal muscle or the adipose tissue cell, and through signal transduction cascades and phosphorylation pathways, you see that the GLUT4 is actually being sequestered from these pools, these intracellular pools of glucose transporter 4, and this signal transduction cascade signals these intracellular pools to exocytose to the plasma membrane and fuse to it, thereby exposing the glucose transporter 4 to the environment, where glucose will then pass through and be phosphorylated by hexokinase 2. Insulin therefore regulates this by binding to the receptor and increasing the amount of glucose transporter 4 on the membrane, but insulin also increases the transcription of hexokinase 2, which we clearly know is coupled to this transporter. Continuing insulin-dependent uptake, there are other forms of regulation of glucose uptake in the muscles and the fat tissue other than insulin, and that is adenosine or GS activators. GS is a GTP coupled protein, and it will activate adenylyl cyclase, which, as you can see, that's usually involved in those signal transduction cascades, and this will increase the glucose transport. On the other hand, lipolytic hormones, isoproterenol, or GI, another GTP protein, activators inhibit adenylyl cyclase and will decrease glucose transport into insulin-dependent uptake, like skeletal muscle. Interestingly, skeletal muscle itself, through contraction, increases glucose uptake. This is through parallel but completely different pathways than insulin-dependent pathways. So it actually has a compounding effect with insulin and glucose uptake. Therefore, exercise can actually be beneficial for people who, like diabetics. Moving on to the liver, again, it is insulin independent, and it is through glucose transporter 2, which is similarly to hexokinase 2 coupled with glucokinase. And what this does is this has an apparatus that forms it's very sensitive to glucose, and it can detect minor changes and subtle changes to blood glucose, which is important for signaling the release of insulin. Glucose phosphorylation is the rate-limiting step in this glucose uptake because glucose transporter 2 has a much higher threshold than glucokinase does for trapping the insulin or trapping the glucose into the cell. Glucose transporter 2, as you know, has a high Km, and so as the blood glucose levels go up, more glucose will be uptaken into the liver cells. To regulate glucose transporter 2, protein kinase C inhibitors like fluoretin and calfostin C will decrease liver glucose uptake because, similarly but a little bit different than glucose transporter 4, those protein kinase C inhibitors will block the translocation of glucose transporter 2 to the plasma membrane. Now we get into the brain. You want to pay close attention to this because this has to do with the tumor cells. Again, just like the liver, this is insulin-independent glucose uptake. You have glucose transporter 1 in the blood-brain barrier, and as well as these astrocytes. Astrocytes are very important at sensing differences in the blood glucose concentration. And you have glucose transporter 3 here, as you can see on the nerve cells, right there and right there. And glucose transporter 3 is throughout the brain and the neurons as well as the central nervous system. And separate from glucose transporter 2, it has a low Km, meaning it has a high affinity for glucose. This is really important because when you have low blood sugar or you don't always have high blood concentrations of glucose, your brain and your neurons are still getting the glucose they need for fuel so that you're constantly able to keep up with all of the synapsing and all of the activity that's going on in your brain all the time. Now, how are these transporters regulated? Well, glucose transporter 1 can be inhibited by fisentin, and as far as I know, this just prevents the glucose uptake across the blood-brain barrier and also in those astrocytes, and this can be very problematic. And also high glucose concentration in the blood downregulates the expression of glucose transporter 1. Glucose transporter 3, on the other hand, is not affected by blood glucose concentrations, and so the expression is always there on the plasma membrane, and that can be very important, again, for low blood sugar levels. You're always getting that glucose to the brain. Needs its energy. However, chronic hypoxia in fetal cells was found to increase glucose transporter 3 mRNA expression, which is pretty interesting. Going back to this picture now that we know about glucose transporter 3 and glucose transporter 1, we have all this glucose in here. So what's going on? It's probably not your blood glucose, meaning it's probably the transporters. There's probably more expression and glucose transporter 3s and some of 1s in the blood-brain barrier, but mostly 3 here around the cells. So the tumors are producing more glucose transporter 3s. Why is this beneficial? Tumors need energy. What's a goal of a tumor? To grow, to spread, sometimes to be hostile to healthy cells and attack those. There's an interesting effect called the Warburg effect, which we actually do see with tumors. They increase their uptake of glucose, but what's interesting is they produce a lot of lactate. And this is the Warburg effect, and what it does is it just makes as much energy, makes as much ATP for these cells as they can make, and it also makes the tumor microenvironment a little acidic, which could be beneficial for the tumor and maybe immunosuppressive, perhaps. There's still a lot that I did not read into this, but it's certainly interesting and goes with the whole tumors want to grow, they want to get energy quick, they don't care how they do it, they're going to produce lactate in the process. Again, so how's this happening? These tumors are upregulating and overexpressing glucose transporter 3 and glucose transporter 1. And there is studies that support this. Glucose transporter 1 and glucose transporter 3 mRNA levels in brain tumors do correspond to the biologic aggressiveness of tumors. So they're saying it makes sense. We are finding increased levels, and it makes sense because they're growing and they need that energy. But what the research does show is that in glioblastomas, it is predominantly glucose transporter 3 that is being upregulated, and they were doing immunorepressive tagging and stuff like that that figured that out. Next case we have is the case of low insulin. So we have a 5-year-old boy. He comes into a doctor's office. Mother said that he's always been sick. He appears drowsy all the time. He's always thirsty and has frequent urination. So they took blood samples, both fasted and after glucose loading, just fed him a lot of sugar, probably a sugary drink, and then they measured his insulin levels. And as you can see here, we're looking at a normal fasting blood insulin of 6 and a control normal of 40 after the glucose load. But the patient only has a blood insulin of 5 after the insulin loading, which is even lower than the fasting control and a really low baseline. So this patient's always insulin deficient. And let's look at the other symptoms again. Drowsy, excessive thirst, frequent urination, low insulin in a fasted and fed state. These are all hallmark signs and symptoms and measurements of type 1 diabetes. Also called juvenile diabetes, so it really makes sense in this scenario. The patient's 5. And so how is this happening? Well, insulin increases glucose uptake into the cells. So the patient is drowsy because the blood glucose is always high. Even in the fasted state, we see here, fasted state, you have hyperglycemia, meaning high blood sugar. These cells are unable to sequester glucose and uptake it, like we talked about earlier in the previous case study. In insulin-dependent uptake, glucose transporter 4 is unable to get to that plasma membrane. These muscle cells, these fat cells, are not getting enough glucose, and so these cells don't have enough energy, so he feels tired. The excessive thirst, that's because your blood glucose, your kidneys get overworked, and it actually pulls more water to excrete it out, so you're always thirsty, and you're drinking a lot of water to rehydrate what you're losing. And you're also peeing a lot because you gotta get rid of that glucose. Now this has a multitude of consequences because we know insulin is a hormone, we know it's one of the key hormones, and it has a lot of effects, both activatory and inhibitory. So again, it's a key hormone for regulation in the human body, especially metabolically. Now, the enzymes that insulin activates, you're not going to have those activated. But on the flip side, the enzymes that insulin deactivates or inhibits, even though you have high blood sugar, you're going to get these other things going on. These enzymes are still active. So glycogen phosphorylase. So one of the main enzymes in glycogenolysis, the breakdown of glycogen stores in your liver and in your muscles, is going to be constantly breaking it down because we're not getting that glucose out of the blood. We got to take our own stores and get energy somehow. Same vein, lipolysis. We got to get energy from our fat stores. This is why you have a lot of diabetic people who are skinny, and who do not have a lot of body fat because you're constantly undergoing lipolysis. Insulin activates a downstream kinase, AKT. AKT inhibits protein kinase A. So if you're not inhibiting protein kinase A, lipolysis occurs. So these are my references for the scholarly articles. And these are my references for